欢迎来到免疫小教室，陪你一起长知识。我是免疫小教室的主持人伊法，将会介绍一系列免疫相关的小教室给大家哦。你知道当受伤的时候，第一个侦测到有细菌入侵到体内的是免疫细胞吗？它们像大楼的管理员一样，只要看到奇怪的人，就会上前去确认身份。每一个管理员身上都有各式各样的受体，可以去侦测不同人的身份，而这些受体我们称之为 PRRs。当这些 PRR 辨识到入侵者，就会透过一系列的反应，号召更多的免疫细胞来抓入侵者。而今天我们要来介绍的是最早被发现的 PRR， 也就是 TLR。想知道 TLR 是怎么去传递讯息给大家知道的吗？就让我们继续看下去吧。
The first step in initiating an innate immune response to infection is the sensing of the pathogens by host cells. This is mediated by the recognition of specific microbial molecules by a class of germline encoded host receptors, which is so-called Pathogen Recognition Receptors, PRRs. PRRs differ in the subcellular location, signaling cascades, and host responses. Based on their subcellular locations, PRRs will be divided into two classes. One class is cell surface PRRs, which include extracellular toll-like receptors and C-type lectin receptors, located in the plasma membrane. They recognize different ligands outside of the cell. The other one class is intracellular PRRs, which include intracellular toll-like receptors, not like receptor proteins, Regi-like receptors, C-gas and related dsDNA sensors located in intracellular compartment. Downstream signaling cascades and host responses of each type of PRR will be introduced afterwards. Among these PRRs, toll-like receptors were the first to be identified and the best characterized. The TLR family comprises 10 members in human and 12 in mouse. TLRs localize to the cell surface or to intracellular compartments such as the ER, endosome, lysosome, and endolysosome, and they recognize distinct or overlapping paths such as lipid, lipoprotein, protein, and nucleic acid. Each TLR is composed of an ectodomain with leucine-rich repeats that mediate PAMP's recognition, a transmembrane domain, and a cytoplasmic TIR domain that initiates downstream signaling. Homo or heterodimer formation initiates signaling to the two major downstream adapter proteins, MyD88 and TRIF. TIRAP conducts the signal from TLR4 to MyD88, and TRAM mediates the signal from TLR4 to TRIF. TLR engagement induces formation of the myddosome, which is based on MyD88 and also contains IRK1 and IRK4. IRK1 activation induces TRIF6 activation following K63 linked polyubiquitination on TRIF6 itself and TAK1. TAK1 activation leads to the activation of IKK complex, NF-κB, and MAPK. MAPK activation leads to AP1's transcription factors activation. TLR engagement also induces TRIF activation following TRIF6 and TRIF3 recruitment. TRIF6 recruits the kinase RIPK1, which in turn interacts with and activates the TAK1 complex, leading to activation of NF-κB and MAPKs and induction of inflammatory cytokines. In contrast, TRIF recruits the IKK-related kinases TBK1 and IKK epsilon for IRF3 phosphorylation. Subsequently, IRF3 forms a dimer and translocates into the nuclease from the cytoplasm, where it induces expression of type I interferon genes. In plasmacytoid DC, TLR7 and TLR9 serve as primary sensors for RNA and DNA viruses. Interestingly, the production of type I interferons by PDCs relies on a complex containing MyD88 and IRF7. This complex also contains TRIF3, TRIF6, IRAK4, IRK1, IKK alpha. Within this complex, IRF7 is phosphorylated by IRK1 or IKK alpha and translocates into the nuclease to regulate the expression of type I interferon. At the same time, recommended bestsellers for TLR signal research from ABclonal are also listed here, including featured antibodies and ELISA kits used for assessment of TLR expression, representative target antibodies for IRAKs, TAK1, TBK1, IKKs, MAPKs, NF-kappa-B, Zhong, and IRFs. For more information of ABclonal products and ordering, please contact our subsidiary in Taiwan. Thanks for your attention.